Hello, my dear students. I hope you all are doing great. My name is Biswajit. I'm your botany mentor. For your KVPY exam, today we are going to discuss a very important chapter of genetics, that is molecular basis of inheritance. And again in this class, we will cover various questions, discuss various questions, and while discussing the questions, I will tell you the related concepts. Okay, so the topics from which we will cover the questions are the DNA, search of genetic material, RNA world, okay, then replication, transcription, even translation. In fact, the entire chapter we are going to cover. Okay, so the class is going to be very important, so please listen to the class very carefully. So the first question is about the structure of DNA, and it says, which of the following statements is correct? Adenine pairs with thymine through 2 hydrogen bond. Yes, this is the correct answer. Okay, why? Because adenine pairs with thymine through 1 hydrogen bond. 1 is incorrect, right? Because it pairs with thymine by 2 hydrogen bonds. Okay, so the answer is going to be A. Answer is going to be A. So what about the hydrogen bond? See, in a double-stranded DNA, we have 2 strands, and these 2 strands are held together by means of hydrogen bonds, right? So the hydrogen bond is present between the nitrogenous bases of the 2 opposite strands, complementary strands. So what is the number of hydrogen bond between the 2 nitrogenous bases? It is dependent on the nature of base pair. If the base pair is 80, the number of hydrogen bond is going to be 2, and if the base pair is GC, the hydrogen bond is going to be 3 in number. One important point which you do not know, let me tell you one thing. DNA is antiparallel in nature. Yes, you are right. But which and if I ask you which bond is responsible for making the DNA antiparallel, then your answer should be, yes, very good, hydrogen bond, not the phosphodiester bond. See, in DNA, the hydrogen bond can be parallel, it can be standard hydrogen bond, or it can be non-standard hydrogen bond. If this hydrogen bond is standard, then the DNA is going to be anti-parallel, and if it is non-standard, then the DNA is going to be parallel. So let me write one thing to you, few things to you that everybody knows this. This is very simple, that between A and T, the number of hydrogen bond is going to be 2, and between G and C, the number of hydrogen bond is going to be C. Everybody knows this, no problem. Now, what about new thing? See, the hydrogen bond between nitrogenous bases, it is of 2 types. What are those? One is standard hydrogen bond, and second is non-standard hydrogen bond, and this non-standard hydrogen bond is also called as Huygens hydrogen bond. Okay, so no problem, non-standard hydrogen bond. Okay, so if the hydrogen bond is standard, then the DNA is going to be anti-parallel. The DNA is going to be anti-parallel DNA, and if it is sorry, non-standard, then the DNA is going to be parallel in nature. DNA is going to be parallel DNA. Okay, and among these 2 types of hydrogen bond, which one is more common? Standard hydrogen bond is more common, and the standard hydrogen bond is also called as Francis-Crick hydrogen bond. This is also called as Francis-Crick hydrogen bond. Okay, now next, purines found in both DNA and RNA. What is purine? Purine is basically nitrogenous base. This is nitrogenous base, and purines, actually in a DNA or RNA nucleic acid, we have 2 types of nitrogenous bases. What are those? One is purine and second is pyrimidine. Okay, so let me write over here that the nitrogenous base is of 2 types. What are those? One is purine and second is pyrimidine. Okay, so what about the purine and pyrimidine? Remember one thing, the purine, it has 2 rings, so I will call this as double ring structure. This is double ring structure, and and how many members are there in its ring? 9 members. So I will call this as 9 membered. This is 9 membered. And what are the examples of purine? One is adenine and second is the guanine. These are the examples of purine. Okay, what about pyrimidine then? This pyrimidine is single ring structure. This is single ring structure. and this has 6 member in its ring. So I will call this as, this is 6 membered. Okay, and what are the examples? The examples are cytosine, thymine, and uracil. And remember one thing, that this thymine is present in DNA. This is present in DNA, whereas this uracil is present in the RNA. This is present in the RNA. But yes, there is an exception. In transfer RNA, there is a loop which is called as T-size loop. And in T-size loop, even though that is a part of tRNA, that is RNA, it has thymine. That is one exception. Okay. See. So this is nitrogenous base, and remember, whether the nitrogenous base is purine or pyrimidine, it is always going to be heterocyclic. So I will write over here that this nitrogenous base is heterocyclic. This is heterocyclic. Why it is heterocyclic? Because the cyclic compound consists of both carbon and nitrogen. Because it consists of both carbon and nitrogen in its ring, that is why this is heterocyclic. And this nitrogenous base is hydrophobic. This is hydrophobic, and because these are hydrophobic in nature, that is why they lie on the inner side of the DNA molecule to avoid the interaction with the external water molecule. Okay, so this was about the nitrogenous bases. Okay, so what is asked in the question? The question says purines found in DNA and RNA, both in DNA and RNA. So the purine which is found both in DNA and RNA, that is the adenine and guanine. So the answer will be adenine and guanine. That means option C. Okay, the association of histone H1 with the nucleosome indicates that. See, so what is nucleosome? Nucleosome is a complex of DNA and proteins, right? So the DNA which is present in the, let me write over here, nucleosome. Why nucleosome has to be formed? Let me tell you one thing. See, in a cell, whether the cell is eukaryotic or prokaryotic, the length of the DNA present in those cells is very, very high as compared to the size of the cell. Okay, and if you talk about the eukaryotic cell, especially the human cell, then the length of the DNA is about 2.2 meter. And to accommodate such a long DNA in such a small cell, we have to pack the DNA, and this packaging of DNA is achieved by the help of histone proteins. Okay, and they are basically basic proteins, positively charged proteins. Okay, so these positively charged proteins, they have positively charged amino acid residues on its surface, and they help to neutralize the negative charge of the DNA molecule. That will help ultimately the coiling or packaging of the DNA molecule. Okay, so the histone proteins are of two types. What are those? One is core histone protein and second is linker histone protein. Okay, so this core histone protein is again of four types: S2A, S2B, S3 and S4. Okay, and two copies of each histone protein, that is S2A, S2B, S3 and S4, they come together to form a octamer, core octamer. Okay, and that is also known as new body, and the DNA gets wrapped around the new body, and once the DNA gets wrapped around the core octamer, then the complex is known as the complex of the core octamer of histone, and the DNA is called as nucleosome. What do you call that? As nucleosome. And why this? And ultimately, so once the nucleosome is formed, the nucleosome is the unit of chromatin. So if the nucleosome is formed, that means it is going to form the chromatin. Okay, and this nucleosome gets associated with the H1, and the exact position of the H1 is still not known. Okay, so if the H1 histone gets associated with the nucleosome, that means what is happening? The DNA is getting condensed to form the what? Chromatin fiber. To form the chromatin fiber. So let me tell you few points about these nucleosomes. Okay, so let me write over here. So in a nucleosome, let me first tell you about the histone proteins. Okay, so the histone protein is basically, what are the properties of histone protein? First, it is basic in nature. When you say basic proteins, that means they are positively charged. They are Why they are positively charged? Because they are rich in positively charged amino acids. Okay, like histidine, right? And then these histone proteins are of two types. What are those? So I will write types. Types. So when you talk about the types, one is called as core histone. One is called as core histone and second is called as linker histone. Second is called as linker histone. Okay. So what about the core histone? The core histone is of four types. What are those? One is S2A and second is S2B. Third is H3 and fourth is H4. These are the core histones. And what about the linker histone? The linker histone is H1. This is the linker histone. Okay. And remember one thing, that when you talk about the presence or occurrence of the histone protein, then histone proteins are present only in the eukaryote, not only in the prokaryote. But this statement is completely not correct. Why? Because if you talk about the prokaryotes, the prokaryotes are basically classified into two groups. What are those? One is archaebacteria. For archaebacteria, I'm writing AB, and second is eubacteria. So I'm writing over here for eubacteria, I'm writing EB, and for archaebacteria, I'm writing AB. Okay. So what about the occurrence of the histone? So the histone proteins are present in the eukaryote, and they are also there, but they are absent in the eubacteria, but they are present in the archaebacteria. So archaebacteria, even though they are prokaryotic in nature, but still they have the histone proteins. This is very, very important. Okay. Now the core histone. So suppose I'm having 8 histones. 2 copies of each of these 4. Okay. So if I take 8 histones over here, so suppose these are the 8 histone molecules. 8 histone molecules. Okay. So 8 histone molecules are there, right? So 8 histone molecules, these 8 histone molecules of what? S2A, S2B, S3 and H4, they are called as what? They are called as core octamer. They are called as core octamer. Core histone octamer. You can call it as core histone octamer. Okay. And this core histone octamer gets wrapped around it by DNA molecule. So here we will have DNA molecule. Okay. So this DNA molecule around the core histone octamer, this is called as core DNA. This is called as core DNA. Okay. And what do you call this core octamer, histone octamer? You call this as new body. Okay. And this core DNA plus new body, that is collectively called as what? Nucleosome. So nucleosome, it does not have H1. Remember one thing, H1 is not a part of nucleosome. This is very, very important. Okay. So I will say which one is not a part of nucleosome. H1 is not a part of nucleosome. Which are present in nucleosome? 2 copies of H2A, 2 copies of S2B, 2 copies of S3, 2 copies of H4, and the core DNA. Okay. So now, in sea urchin DNA, which is double stranded, 70% of bases were shown to be cytosine. The percentages of the other 3 bases expected to be present in. See, this is a question which is based on the DNA structure, and we can solve it with the help of what? With the help of Chargaff's rule. What does this Chargaff rule say? Chargaff's rule says that in a double stranded DNA, the amount of purine is equals to the amount of pyrimidine. So I will say the total amount of purine is equals to the total amount of pyrimidine. So what are purine? So the purine are adenine and guanine. So I will say adenine plus guanine is equals to cytosine plus thymine. Okay. So I can also write here that A plus G upon C plus T, it will be 1. It will be always 1. Okay. And see, if we know that in DNA only 4 different types of nitrogenous bases are there. So I can say that A plus G plus C plus T is equals to 100%. is equals to 100%, and because A plus G is equals to C plus T, so I can say that I can write over here that A plus G will be 50% and C plus T will be 50%. C plus T is 50%. And this Chargaff's rule also says that, the amount of adenine in a DNA is equals to amount of thymine, and the amount of guanine in the DNA is equals to that of the amount of cytosine. Okay. So this is about the number. It is talking about the number, not the hydrogen bond. Okay. So the question says that the 70% of bases are cytosine. 17% is cytosine. So I will write over here. So I can write over here the solution. In this DNA, because this DNA is double stranded and Chargaff's rule is applicable only for double stranded DNA. Okay. So as per the question, the given data says that cytosine is 17%. Cytosine is 17%. So if cytosine is 17%, then obviously then the guanine will also be 17%. Why? Because the amount of G is equals to amount of C. Okay, and we know that A plus G is 50%. A plus G is 50%. So because G is 17%, so I will say that A will be 33%. A will be 33%, and because amount of A is equals to amount of T, that is why the amount of T will also be 33%. It will be 33%. Okay. So I can write over here that G is 7%. Yes, it is right. Okay. A and T, they are 33%. So the answer will be A. Okay. So the thing which you have to remember is, in this case, A plus G is 50% and C plus T is 50%. Okay. This is very simple. Now next question. The diagram shows the important concept in the genetic implication of DNA. Fill in the blanks. See, the information in the DNA is present in the form of base pairs. Okay, and this base pair, the information present in the form of base pair, it has to be transferred to form the proteins. So how does this happen? Initially, the DNA is copied to form the RNA molecule, mRNA molecule. Okay, now in the mRNA, the genetic information is present in the form of nucleotide sequence or base sequence, not base pair. In DNA, this information is present in the form of base pair sequence, and in RNA, it is present in the form of base sequence or nucleotide sequences. And in mRNA, we say that the information is present not only in the form of nucleotide or base sequences, we also call it as codons. And what is codon? Codon is basically collection of 3 adjacent nucleotides which code for certain amino acids, or it may not code for amino acid, which are called as stop codons, right? See, so how does this happen? The DNA which has the information, it is initially, it transfers its information to the RNA. Okay, so we are having DNA molecule, and the information present in the DNA molecule is transferred to the mRNA molecule or RNA, any RNA molecule. And the process in which DNA forms RNA, any type of RNA, mRNA, rRNA, tRNA, does not matter. If it forms RNA molecule, then the process is going to be called as transcription. This process is going to be called as transcription. So this process is transcription. Okay, and the mRNA, it has codons, and the codons are read by the ribosomes, and it ultimately forms the polypeptide or proteins, and this process is called as translation. Okay, and this process, you can see over here, the DNA is getting converted into DNA, and this process is called as replication. What do you call this as? Replication. Okay, and this flow of information, that is from DNA to mRNA and from mRNA to protein, this is called as central dogma. Unidirectional central dogma, and this was given by Francis Crick. Who gave it? Francis Crick gave this central dogma. So A is transcription, B is translation, and C is Francis Crick. So the answer is A. Okay, now what are the structures called that give an appearance as beads on string? Okay, so this is an typing error, no issues. So it is actually beads. Okay, beads on a string. So this is basically the nucleosome. What is that? Nucleosome. So the presence of the nucleosome gives beads on string-like appearance. Beads on string-like appearance. Okay, so whenever beads like string appearance is given, you have to think about the nucleosome. And in this case, the beads. In this case, the beads are basically nucleosomes. The beads are basically nucleosomes, and this string is basically compared with DNA molecule. Compared with DNA molecule. Okay. Now, which one of the following does not follow central dogma of molecular biology? See, in normal central dogma, what happens? The information present in the DNA, that is transferred to the RNA molecule, especially the mRNA molecule, and from mRNA molecule, the information is transferred to the protein molecule. Okay, so this is applicable to the cellular organism. So what is cellular organism? The organism which has cell. But if you talk about the HIV, HIV is a virus. What type of virus? RNA virus, retrovirus. Okay, and in this HIV virus, the genetic material is RNA. And what type of RNA it is? The genetic material is single-stranded RNA molecule. And because it has single-stranded RNA molecule, that is why it is called as what? Retrovirus. It is called as retrovirus. So in this case, normal central dogma does not take place. So I will say that this is an exception. Okay. And see, in HIV virus, we have two copies. Of single-stranded RNA. molecules. Okay. Now, the 3' to 5' phosphoanhydride linkages inside the polynucleotide chain serves to join what? One nucleotide with the another nucleotide. One nucleoside. No, not nucleoside. This nucleotide. So one nucleotide with the another nucleotide. So this is going to be the answer. One nucleotide with the another nucleotide. Okay. So let me tell you one important point, that what are the different linkages in a DNA molecule? So first, let us talk about the nucleotide. Okay. So in a nucleotide, what are the bonds? Let me just check the time. Okay, so I'm going to tell you about some of the important linkages in a nucleotide. Okay, so I'm writing over here linkages in a nucleotide. The nucleotide can be deoxyribonucleotide or ribonucleotide. It does not matter. I'm simply writing nucleotide. Okay, so here I'm simply drawing a sugar. And the sugar which is present in the nucleotide is 5 carbon. That means it is pentose sugar. So let me draw one pentose sugar over here. So suppose this is pentose sugar. And this pentose sugar is, again, it is going to be what? It is going to be heterocyclic. Why? Because it has oxygen and in other places it has carbon. Okay, so let me draw the carbon over here. So this is 1 carbon, this is 2nd carbon, this is 3rd carbon, this is 4th carbon, and I will also have another carbon. So this is 5th carbon, and these carbons are named as prime. So this is 1' carbon, 2' carbon, 3' carbon, 4' carbon, and 5' carbon. Why do we call these carbons as prime? Because in the nucleotide we also have nitrogen, and nitrogen also has carbon. So nitrogenous base, and nitrogenous base also has carbon. So to distinguish whether the carbon belongs to the nitrogenous base or the sugar, we name these carbon as prime in case of sugar, and we simply name them as 1 or 2 or 3 in case of nitrogenous base, just to distinguish them. Okay, so these are the carbon molecules, these are the carbon atoms. Okay, so these are the carbon atoms, right? So here I will have what? Hydrogen and OH groups. Okay, so suppose if the nucleotide is ribose sugar, so let me draw one ribose sugar. So here I will have OH, here I will have H, here I will have H, and this is OH. This is going to be OH. Here I will have the H, and here I will have OH, and here I will have H group. Okay, and in this case I will have CH2OH. right? So this is a ribose sugar. This is a ribose sugar. Okay, and now this ribose sugar is going to be bonded with the nitrogenous base. Suppose the nitrogenous base is pyrimidine. What is this? That is pyrimidine, and pyrimidine has single ring structure. So let me draw one single ring structure. Just a minute. So this is a nitrogenous base. Okay, and suppose the nitrogenous base is, so this nitrogenous base is pyrimidine. Okay, what is this? Nitrogenous base. And in this nitrogenous base, I will have nitrogen. So here I'm having nitrogen, and this nitrogen is bonded with the hydrogen. This is bonded with the hydrogen. Okay, so this is a sugar and this is the nitrogenous base, and these two molecules, they will get bonded with each other. So in that case, what will happen? These two atoms, these two groups, that is the hydrogen and OH, they will interact with each other, and finally H2O will get removed. Okay, what is going to be removed? H2O. H2O will get removed, and when the H2O gets removed, one bond will be formed between the nitrogen and the carbon. Okay, and this bond is called as glycosidic bond. Which bond? Glycosidic bond. So this is basically glycosidic bond, and this sugar had OH group above the ring, and when this sugar has OH group at 1' carbon above the ring, that is called as beta sugar. What do you call that? As beta form of sugar. So that is why the glycosidic bond is called as beta glycosidic bond. Beta glycosidic bond or linkage, you can call it as beta glycosidic bond or linkage. Okay, and in this glycosidic linkage, you can see that which atom is involved? Nitrogen is involved. That is why we call this as beta N glycosidic linkage. What do you call that? As beta N glycosidic linkage. So one thing you have to remember that in a nucleoside we have sugar and nitrogenous base, and between sugar and nitrogenous base, which bond is present? Beta and glycosidic bond is present. Now I have to draw I have to draw the phosphates. So suppose this is the phosphate. This is the phosphate. Okay, now these two OH, they will interact with each other, and when they interact with each other, what will happen? H2O will get removed. What will get removed? H2O will get removed, and when the H2O gets removed, what will happen? One bond will be formed. So what will be the bond? Let me write that over here. Okay, so here I'm writing that OH like this. Okay, so nitrogenous base plus sugar, nucleoside, and here I'm having phosphate, so this is known as nucleoside monophosphate, ribonucleoside monophosphate, right? And if I want to join other phosphates, so I can also join them over here, so, and that will be like this: oxygen, phosphorus, oxygen, phosphorus, OH group. Okay, P double bond O, P double bond O, and here I will have OH, and here I will have OH like this. And this, so here you can see that we have three phosphate groups. The phosphate group which is attached to the 5' carbon, that is called as alpha phosphate. Which phosphate? Alpha phosphate. And this phosphate is known as beta phosphate, and this phosphate is known as gamma phosphate. Which phosphate? Gamma phosphate, right? And so what about the linkages? What about the linkages between the sugar and the first phosphate, that is the alpha phosphate? So this linkage is basically phosphoester linkage. Which linkage? Phosphoester linkage. Why? Because in ester linkage, we know that the ester linkage consists of what? C double bond O, oxygen. This is the ester linkage. Okay, and if instead of carbon, if I write over here phosphorus, then the linkage will be phosphoester linkage. Which linkage? Phosphoester linkage. Okay, so this is phosphoester linkage. And what about these linkages? That means this one and this one. Sorry. What about these two linkages? Okay, these two linkages are basically phosphoanhydride linkage. Why? Because see, I'm writing over here C double bond O, oxygen. This is ester linkage. What is this? This is ester linkage. So I will write P double bond O oxygen. This will be phosphoester. This will be phosphoester linkage, right? And then if I write over here C double bond O oxygen, C double bond O, okay, this is anhydride linkage. This is anhydride linkage. Okay, so instead of carbon, if I write P over here, so this will be phosphoanhydride. This will be phosphoanhydride, right? So in these cases, you can see P double bond O oxygen, P double bond O oxygen. Here also P double bond oxygen, P double bond O oxygen. So so here I can say that these linkages are basically what? Basically phosphoanhydride linkages. So I will say one thing, So from here, one thing you came to know that in case of a nucleotide, the linkage between the alpha phosphate and the carbon is what? Phosphoanhydride. What is that? Phosphoanhydride. And between the alpha and beta, phosphoester, between alpha beta phosphoanhydride, and between beta and gamma, it is again phosphoanhydride linkage. Okay, so these are the linkages in the nucleotide. So what about the linkages in a DNA molecule? Let me tell you one by one. So if you talk about a DNA molecule or RNA molecule, it does not matter. So see, in a nucleotide, I'm writing over here what? Linkages in a polynucleotide chain. So I'm writing over here linkages in a polynucleotide chain. Polynucleotide chain. Polynucleotide chain. Okay, so let me draw a diagram over here. So suppose this is a sequence. Okay, and here I'm having so suppose this is 5 prime end. Suppose this is 5 prime end. Okay, now let me draw over here. Suppose this is CH2 5 prime carbon. Okay, and this is the sugar. This is the sugar, and the sugar can belong to any may belong to ribose or deoxyribose. It does not matter. It does not matter. Okay, and here I will have the nitrogenous base. I will have nitrogenous base. This is the sugar. Okay, this is 3 prime carbon. This is 2 prime carbon. This is 1 prime carbon. Okay, and here I will have hydrogen. And this 3 prime carbon is associated with what? Here I will have oxygen. Okay, and here I will have phosphorus, P double bond O. Okay, and here I will have OH group. And here I will have oxygen, and here I will have CH2 of other sugar, and here I will have what? Here I will have sugar over here. So this is the sugar. And here I will have nitrogenous base. Okay, so this is the structure of what? A polynucleotide chain. So here I am having, this is a polynucleotide chain. This is the 5 prime end, and this is what? This is the 3 prime carbon. And here this is 3 prime carbon, and here I will have hydroxyl group. So this end will be called as 3 prime end, and this end will be called as 5 prime end. Okay, so here you can see this is what? This is a sugar molecule. Okay, and this is what? This is nitrogenous base, and here I will have what? Phosphate group. So this will be called as one nucleotide, and this is also one nucleotide. This is also called as one nucleotide. Okay, so between these two nucleotides, which linkage is present? This linkage is present. And this linkage is called as phosphodiester linkage. Why? Because see, if I say P double bond O oxygen P, so this is basically phosphoester linkage. This is phosphoester linkage. So here you can see that this is one phosphoester linkage. And this one is also a phosphoester linkage. So how many phosphoester linkages are there? 2. So that is why I will call this as what? Phosphodiesterlinkage. What do I call that? This as phosphodiester linkage. So I will write over here phosphodiester linkage. And in this phosphodiester linkage, let us learn about the carbon. Okay, so this is 3 prime carbon, and this is 5 prime carbon. So this phosphodiester bond is formed between the 3 prime carbon and 5 prime carbon. So I will write over here that this is 3 prime to 5 prime phosphodiester linkage. 3 prime to 5 prime phosphodiester linkage. And which bond is this one? This is the beta N glycosidic linkage. This is beta N glycosidic linkage. Okay, so these two types of linkages are there in a polynucleotide chain. One is beta N glycosidic linkage and second is the phosphodiester linkage. Phosphodiester linkage. Okay, so now let me tell you about other linkages in a polynucleotide chain. Okay, or suppose the DNA molecule, double stranded DNA molecule. So suppose I'm writing over here a polynucleotide chain. So here suppose I'm having phosphate, so the P is phosphate, not phosphorus. Okay, then I am having sugar, then I'm having what? Then I'm having phosphate, and again I'm having sugar, and I'm having phosphate, and I'm having sugar, and this sugar at the its 3 prime end, it will have what? It will have OH group. It will have OH group. So this is and apart from this I will also have nitrogenous bases. So this is the nitrogenous base, and this nitrogenous base is conjugated with what? Sugar. So these are the nitrogenous bases. These are the nitrogenous bases. So this is a polynucleotide chain. This is a polynucleotide chain. Okay. So actually here I am having 3 polynucleotides. So this is actually a diagram of what? Trinucleotide chain. So if I draw a huge number of or numerous nucleotides, then it will be polynucleotide chain. Okay. So in this case, I will say that this is this one. Sorry. This one is called as backbone of the nucleotide chain. This is called as backbone of the nucleotide chain, and the backbone of nucleotide chain consists of what? Alternate phosphate and sugar. Alternate phosphate and sugar. And this contains what? Phosphodiester linkage between the two sugars of two adjacent nucleotides. Which bond will be there? Phosphodiester bond will be there. Okay. Now I can also draw. I can draw another diagram like this. So here I will have nitrogenous base. Okay, here also I will have nitrogenous base, and here also I will have nitrogenous base. Okay, and these nitrogenous bases will be conjugated or bound with the what? Bound with the sugar molecules. So here I will have sugar molecules. Okay, and here I will have phosphate. Okay, and here I will have phosphate. Okay, and here I will have OH group. So the end of the nucleotide chain which has OH group, that end will be called as what? 3 prime end, and this end will be called as 5 prime end, right? And here this one is again the backbone of what? Another nucleotide chain. Okay, and in this case suppose the nucleotide chain, suppose this nitrogenous base is A, so here it will be T. If it is C, it will be G. If it is T, it will be A. And if the nitrogenous base pair is Ta, then the hydrogen bond will be 2 in number. And if it is G and C, the hydrogen bond will be 3 in number. 3 hydrogen bonds will be there. Here I will have 2 hydrogen bonds. Okay, so the 2 strands are held together by what? Hydrogen bonds. Between what? Between the nitrogenous bases, and these nitrogenous bases are said to be complementary to each other. Okay, so these are the bonds or linkages. So in a double stranded nucleotide, what are the linkages present? Here this one is the phosphodiester linkage, this one is the beta N glycosidic linkage, and this one is called as what? This one is called as hydrogen bond. And apart from hydrogen bond, the DNA is stabilized by another force that is called as stacking force. And what is stacking force? This is one base pair, this is another base pair. Between these two base pairs, a force of interaction takes place, and that is called as stacking force. And this stacking force is the major force which is responsible for stabilizing the what? DNA molecule. Okay, and this stacking force is what? Sequence dependent. This is sequence dependent. That means if the base pair is GC, the stacking force will be more, and if it is AT, it will be less in number, less in amount. So I will say that what is stacking force? Let me write over here. Stacking force, you can write over here. You please write. Stacking force is what? The force of interaction between two adjacent base pairs. Okay, and second, it is the major force for stabilizing the DNA. Okay, not the hydrogen bond. This is the what? Stacking force, which is the major force for stabilizing the DNA molecules. And then this is sequence dependent. That means what will be the amount of stacking force? It will depend upon the what? It will depend upon the base pair sequences. So let me write over here. So if the base pair is GC, if one base pair is GC and second base pair is GC, then the shaking force will be maximum. And if the one base pair is GC and second base pair is AT, then shaking force will be relatively less. And if the base pair is AT and another base pair is AT, then the shaking force will be minimum. So this is the, so you can see that this shaking force is dependent upon the base pairs. Okay, GC base pair, that then this shaking force will be maximum. AT then this shaking force will be minimum. Okay, so these are the different structure, the different linkages in a what? In a DNA or any nucleotide chain. Okay, now let's move to the next topic. Sorry. Which one of the following statements about the particular entity is true? Centromere is found in animal cell which produces esters during division. Yes, centromere is found in the animal cell, no problem. But centromere is a part of what? This is a part of chromosome, and chromosome does not produce ester. So it should have been centriole. So this is incorrect statement. This is incorrect. We have to find out the true statement. So this is false statement. The genes for producing insulin is present in every body cell. This is true statement. So the answer is B. Why? Because here we do not have more than one option. So the answer is B. Nucleosome is formed by nucleotides. No, nucleosome is formed of what? We have nucleosome is formed of what? New body, which is called as core histone octamer plus core DNA. These two components, they form the nucleosome. DNA consists of core histone octamer. No, it is the it is the new body. It is the new body or core histone octamer that consists of what? 8 histone octamer. So this option is incorrect. So the answer is B. So you may ask one thing. So let me explain you this part. This is very important. See, every cell of an organism has same genes. That means the cell of my skin, the genes which are present in the cell of my skin are exactly identical to the genes which are present in the liver, in my liver cells. Okay, that means every cell of an organism, of the body of an organism, have same set of genes. So you may wonder one thing, that if the skin cell has the same gene as that of the beta cells, then why the skin cell does not produce the what? Does not produce the insulin. Why? Because of the differential transcription. Because of the differential transcription. See, every cell has same genes, same set of genes, but in some cells some genes will be expressed, and in some cells some other genes will be expressed. Why? Because of the differential expression of transcription factor. Let me explain you one point over here. So suppose this is one cell. Suppose this is one cell and this is another cell. This is another cell. And suppose this is beta cell. Suppose this is beta cell, and suppose this is immature RBC. So immature RBC has nucleus, and if it has nucleus, then it will produce the it will have DNA. That means it will have genes. Okay, so let me write over here some of the genes. Okay, so suppose this is one gene. Okay, and these, because these genes are same genes are present everywhere, so genes are present. Okay, so this is one gene. Okay, and let me draw another gene over here. Another gene over here. Okay, and so these are the different genes. Okay, these are the different genes. So suppose every gene which undergoes transcription, it will have promoter. So suppose this is the promoter area, this is the promoter area, this is the promoter area, this is the promoter area. So these are the promoters. And remember one thing, during transcription, a number of proteins they get bound to the promoter, and they are said to be transcription factor. Before binding of the RNA polymerase to the promoter, certain factors they get bound to the promoter, and they are said to be transcription factor. And different genes have different promoters, and different promoters get bound to the different transcription factors. Okay, same transcription factor cannot bind to the different promoters. Every promoter has its own transcription factor. So this is the promoter, these are the different promoters. Okay, so suppose this is the gene which is responsible for producing phosphofructokinase, and this is an enzyme which is involved in the glycolysis, and this is the same enzyme phosphofructokinase. And suppose this is a gene which is responsible for forming insulin, so this is insulin forming gene. Okay, and this one is alpha chain forming gene, and this is beta chain forming gene. So this is what? This is insulin forming gene, this is alpha chain forming gene, and this is beta chain forming gene. So you can see all the genes are present in both beta cell and what? RBC. Now, so this is the promoter, and this promoter gets bound with the transcription factor. So suppose for phosphofructokinase, the transcription factor is 1. Okay, and for insulin, suppose the transcription factor, this transcription factor is 2, and for alpha chain producing promoter, this is 3, and for beta chain producing, it is suppose 4. It is 4. So in this case, what happens? Transcription factor 1 is produced. So if the transcription factor 1 is produced, so it will get bound to this here over. Okay, so if this transcription factor 1 binds to the promoter, then it will undergo transcription, and it will ultimately form the protein, which is what? Phosphofructokinase. So in this case, phosphofructokinase is produced. Okay, and in this case, in beta cell, the transcription factor 2 is also formed. So it will get bound to the promoter, and when the transcription factor gets bound to the promoter, ultimately it will result in the transcription. So after transcription, translation will take place and insulin will be formed. So here insulin is formed. But in this case, transcription factor 3 is not formed. In this case, transcription factor 4 is not formed. And because these are not formed, that is why they will not get bound to, that means the promoter will not get bound to the transcription factor, and that is why there will be no transcription of this gene and this gene, and that is why there will be no formation of what? Alpha chain and beta chain. So in this case, I will say that this cell is producing phosphofructokinase and insulin. Now in this case, again, the transcription factor 1 will be produced, so it will bind to the promoter, and it will produce the phosphofructokinase ultimately. And in this case, the transcription factor 2 is not formed, so insulin will not be formed. But in this case, transcription factor 3 will be formed, so it will bind to the promoter, and hence that is why alpha chain will be formed. And transcription factor 4 is also formed, so it will get bound to the promoter, so beta chain will be formed. Alpha chain and beta chain, when they are formed, that will form the formation of the hemoglobin. So in this case, you can see that when you compare these two cells, RBC and beta cell, phosphofructokinase is formed in both cases. So such genes which are expressed in all the cells, they are said to be constitutive genes or housekeeping genes. Constitutive genes or housekeeping genes. And if you talk about insulin gene or hemoglobin gene, you can see that insulin is produced in beta cell, whereas it is not produced in RBC. But hemoglobin is produced in RBC, and hemoglobin is not produced in the beta cell. So why this cell is beta cell, why this cell is RBC? You can see that gene is same, the set of gene is same over here, but the product is different. Why? Because of the differential expression of the transcription factors. You can see the transcription factors present in this cell are not identical to the transcription factors present in this cell. Yes, similar. Here we have transcription factor 1. Here also I am having transcription factor 1. But in this case, I have transcription factor 2. But here I do not have transcription factor 2. Here I do not have 3 and 4. Here I have 3 and 4. So I will say that the cell differentiation, why it became beta cell? Because it produced the insulin. Why it produced insulin? Because of the production of transcription factor 2. Why it became RBC? Because it produced hemoglobin. Why it produced hemoglobin? Because of the expression of the transcription factor 3 and 4. So I will say, and this formation of different types of cells is called as cell differentiation, and the cell differentiation is due to the differential expression of transcription factor. So I will say one point over here, that cell differentiation this is due to differential expression of transcription factors. Differential expression of transcription factors. Okay, let me check the time first. Now, okay, let's move to the next question. The next question says that in DNA molecule, the proportion of adenine in relation to thymine varies from with organism to organism. No. In every organism, if the DNA is double stranded, as per Chargaff's rule, the amount of adenine will always be equals to the amount of thymine. If the DNA is double stranded, in every double stranded DNA, amount of adenine will be equals to amount of thymine. Okay, so it is saying that varies, and because it is saying varies, so this is incorrect. There are two strands which run anti-parallel. One strand is 5' to 3', other is 3' to 5'. This is correct statement. Okay. The total amount of purine nucleotides and pyrimidine nucleotides is not always equal. No, it is always going to be equal. Why? Because purine is equals to pyrimidine if the DNA is double stranded. If the DNA is double stranded, then the amount of purine will be amount of pyrimidine. There are two strands which run parallel. Okay, see, normally the DNA is anti-parallel. So this is incorrect. So the answer is B. Okay, next. Which one of the following pairs of nitrogenous bases of nucleic acid is wrongly matched with category mentioned against it? See, we have two groups of nitrogenous bases, purine and pyrimidine, right? So in case of purine, as I have already told you, in case of purine, I have adenine and guanine, and in case of pyrimidine, I have cytosine, thymine and uracil, right? So guanine and adenine, they are purine, and here the question says wrongly matched. This is true, this is correct match, so this won't be answer. Adenine and thymine, they are purine. No, thymine is not purine, so this is incorrect. So this will be the answer. Okay, this is purine and this is pyrimidine. So this will be the answer. Thymine and uracil, they are pyrimidine. This is true match. Okay, and uracil, cytosine, they are pyrimidine. This is also correct. So the answer will be B. Next, 1 turn of helix of B form of DNA is approximately. So when the DNA, we know that DNA is helical in nature. So 1 complete turn, when the DNA undergoes 1 complete turn, that means when it coils for 360 degree. One complete turn, that means 360 degree. The length of the DNA, the rise of the helix is about what? Is about 3.4 nanometer, or I can say 34 angstrom. And 1 complete turn of the DNA helix contains 10 base pairs. So I will write over here that one turn of the helix, it contains 10 base pairs. It contains 10 base pairs, and between two base pairs, the distance between two base pairs is 3.4 angstrom. And because we have 10 base pairs, so the total length will be 10 into 3.4 angstrom. So the answer will be what? 34 angstrom. And this 34 angstrom, when it is converted into nanometer, so it will be 3.4 nanometer. So the answer will be 3.4 nanometer. Next, anti-parallel strands of DNA molecule means one strand turns clockwise, one strand turns anti-clockwise. The phosphate groups of two strands at their ends share the same position. The phosphate groups at their groups at the start of the two strands are opposite in position or opposite pole. So the answer is D. Why? Because if I draw a diagram of what? DNA. So suppose this is one strand and this is another strand. So in this strand, phosphate will be present and at this end, and here the phosphate will be present at this end. Here the OH will be present over here, and here the OH will be present over here. So this is 5' and this is 3' and this is 5' and this is 3'. So you can see at two different poles phosphates are present. So the answer is D. Next, which one of the following makes the use of RNA template to synthesize DNA? Okay, so we are having DNA polymerase. So DNA polymerase is basically DNA dependent DNA polymerase. So in this case, the template is The template used by the DNA polymerase. The template used by the DNA polymerase is DNA. Okay. RNA polymerase, it uses again the template used by the RNA polymerase is RNA. Sorry, DNA. It is DNA. Okay, see, DNA polymerase synthesizes DNA. So this is called as DNA polymerase. Okay, and it uses DNA as template. So I will call this as DNA dependent DNA polymerase. Okay, RNA polymerase, it synthesizes RNA. So it is called as RNA polymerase, and it uses DNA as template. So it is called as DNA dependent RNA polymerase. DNA dependent RNA polymerase is basically RNA polymerase. So it is going to use what? It is going to use DNA as template. What about reverse transcriptase? Reverse transcriptase synthesizes DNA. So it is called as DNA polymerase. It is called as DNA polymerase. Which one? Reverse transcriptase. But it uses RNA as template. So I will call this as RNA dependent. This is RNA dependent DNA polymerase. Okay. So this reverse transcriptase uses which one as template? RNA as template. So this is the answer. This is the answer. Okay, DNA polymerase means when it is simply written DNA polymerase, that means it is DNA dependent DNA polymerase. Okay, so when RNA dependent DNA polymerase is given, that means you have to consider reverse transcriptase. Okay, now next. Which one of the following hydrolyzes internal phosphodiester bond? Internal phosphodiester bond. Lipase, no. Phosphodiester bond is not broken by lipase. Okay, protease not. What? It is the endonuclease. It is the endonuclease. So what is endonuclease? What is exonuclease? Let me tell you one by one. Okay, so suppose these are the nucleotides. These are the nucleotides. Okay, and between these nucleotides, I will have phosphodiester linkages. I will have phosphodiester linkages. Okay, so suppose this is 5' and this is 3'. This is 3', and these phosphodiester linkages are classified into two groups. Okay, this phosphodiester linkage and this phosphodiester linkage, they are called as terminal phosphodiester linkage. Terminal phosphodiester linkage, and this is also terminal phosphodiester linkage. Terminal phosphodiester linkage. Okay, and the remaining are said to be what? These are called as internal phosphodiester linkages. Internal phosphodiester linkages. Okay, and these linkages are phosphodiester linkages present in where? In the nucleic acid. In the nucleic acid, right? And when the nucleic acid is broken down, that means in that case phosphodiester linkage is broken down. So the enzyme will be called as nuclease. The enzyme will be called as nuclease. Now, if you break this terminal phosphodiester bond, then this nuclease will be called as exonuclease. That will be called as exonuclease. So this will be exonuclease. Exonuclease. Okay, and if you remove, if you are removing one nucleotide from the 5' end, then the exonuclease will be called as 5' to 3' exonuclease. 5' to 3' exonuclease. And if you are removing nucleotide from the 3' end, that means if you are removing terminal phosphodiester bond from the 3' end, that will be called as what? 3' to 5' exonuclease activity. And in case if you break these phosphodiester bonds, then that enzyme will be called as endonuclease. Endonuclease. So endonuclease, what do? They actually break the internal phosphodiester bond. And there are certain enzymes which break certain internal phosphodiester bonds. They are said to be restriction endonuclease. So restriction endonuclease is a special type of what? Endonuclease. Okay, now next. The following ratio is generally constant for a given species. Okay, generally, it is saying that generally, which ratio is generally constant? So we know that if I talk about adenine plus guanine upon cytosine plus thymine, it is always 1. It is always 1 for every organism. But if I take another example, that is A plus T upon G plus C, this is not 1. This is not 1, and the value of this ratio is species specific. This is species specific. That means in one species the ratio is always going to be same. Okay, so A plus G plus C plus T, it is here the word generally is given, so generally is given, so this will not be the answer. Okay, because it is always 1. T plus C upon G plus A, T plus C upon G plus A, this one is the okay, this one is always going to be 1, so this is not the answer. Okay, G plus C upon T plus A, yes, this is the answer. A plus C plus T plus G, no, this is not the answer, so the answer is C. The answer is C. Now next, just a minute. Okay, next, in a DNA, percentage of thymine is 20%. What is the percentage of guanine? What is the percentage of guanine? See, as I have told you, as per the Chargaff's rule, the DNA is double stranded. If the DNA is double stranded, Chargaff's rule can be applied, and this says that the total purine is always 50%. Always 50%. Okay, and as per the question, the thymine is 20%. Okay, so the pyrimidine will be what? Pyrimidine will also be 50%. So I will write over C plus T is 50%. C plus T will be 50%. So thymine is 20%. So the thymine is 20%. Thymine is 20%, and we know that adenine, amount of adenine is equals to amount of thymine. So here, okay. So thymine is what? Thymine is 20%. Thymine is 20%. So if thymine is 20%, then cytosine will be what? 30%. Cytosine will be 30%, right? So if cytosine is 30%, then guanine will be what? Guanine will be 30%. So that is why the guanine will also be 30%. Why? Because the amount, we know that amount of guanine is always equals to amount of cytosine. Always equals to cytosine. So you can see thymine, if thymine is 20%, then adenine will be 20%. Adenine will be 20%. Adenine will be 20%. Okay, and in this question, what is asked? Guanine is asked. So if adenine is 20%, the guanine will be what? 30%. Guanine will be 30%. Why? Because adenine plus guanine is 50%. So the answer will be guanine is 30%. So the answer is C. Next, DNA is mainly found in nucleus. See, there is a small typing error. Nucleus and cytoplasm. See, the DNA is found in both nucleus and cytoplasm. Where in cytoplasm? In cytoplasm we have organelles like mitochondria and what? Mitochondria and what? Plastid. All the three types of plastids, they have DNA. So the answer is B. Okay, in nucleolus we do not have DNA. In prokaryotes the genetic material is DNA. What type of DNA? Circular DNA. Okay, and this is not generally associated with histone. Why? Because when you talk about prokaryote, you have to consider with respect to eubacteria, not with respect to archaebacteria. Archaebacteria, they do have histone proteins, but you have to, while solving questions, you have to consider eubacteria. Okay, so it is circular without DNA, without histone proteins. Next, DNA when AGCT occur, the association as per the following pair. So we know that A pairs with what? A pairs with thymine and G pairs with cytosine. Okay, G pairs with cytosine. So the answer will be ATGC. A will pair with T and G will pair with C. Next, genes are packed into bacterial chromosome by. See, in bacteria, the nucleoid is called as bacterial chromosome. So in nucleosome, bacterial chromosome consists of, the bacterial chromosome is basically nucleoid. This is basically nucleoid. Okay, and this nucleoid or bacterial chromosome consists of DNA. What type of DNA? One circular double stranded DNA is present. Okay, and the DNA for its packaging, it requires proteins, and those proteins are basically basic proteins. That means positively charged basic proteins, and these proteins are not histone proteins. And the examples of basic proteins are basically polyamines. And the DNA which is present in the what in the bacterial chromosome, they undergo transcription to produce the RNA molecules. So few RNA molecules will be there. Okay, so I will say the bacterial chromosome, that is the nucleate, consists of one double-stranded circular DNA and basic proteins, a number of basic proteins, and it will have few RNA molecules. Okay, so for packaging of the bacterial chromosome, what is required? The basic proteins are required. Which are required? Basic proteins. Why? Because these basic proteins are required for packaging of DNA. They are meant for packaging of DNA. Okay, next. Nucleosome core is made of. Nucleosome core is known as what? Nucleosome core is known as histone octamer. It is called as histone octamer. Okay, or you can also call this as new body. And it consists of two copies of each S2A, S2B, H3 and H4. They will be present. Okay, so the answer will be 4. The answer will be 4. Right? Next. DNA with unequal nitrogenous bases are mostly, okay. So if the DNA has unequal nitrogenous bases, that means the amount of adenine is not equals to amount of thymine, and if the amount of guanine is not equals to cytosine, then the DNA, that means this is not following the Chargas rule. And when the Chargas rule is not followed, that means the DNA is single stranded. So I will say this DNA is what? Single stranded. The DNA is single stranded. Next. An octamer of 4 histones complex with DNA forms. What? That is called as nucleosome. We have already discussed this one. Next. The experimental proof for semi-conservative replication of DNA was first shown in. See, the semi-conservative mode of DNA replication was first proved by what? By whom? Michelson and Stoll. These were the two scientists who for the first time experimentally proved that okay, the DNA replication is semi-conservative, and they experimented in the organism E. coli. Where did they experimented? In E. coli. And what is this E. coli? This is a bacterium. So the answer is going to be B. And Michelson and Stoll, they used what? They used cesium chloride density gradient centrifugation. And they supplied nitrogen in the form of NH4Cl. And this NH4Cl, in NH4Cl, the nitrogen supplied were either in the form of heavy isotope or light isotope. That means it was N15 and N14. In these two forms, the nitrogen was supplied to the culture medium. Okay, so the answer is B over here. Next, select the correct match. Ribosome. What is ribosome? Ribosome. See, the enzymes. When you talk about the enzymes, they are biocatalysts, and they are always positive catalysts. Why? Because they always increase the rate of reaction. Okay. Now, these enzymes, when we talk about the chemical nature of the enzymes, these enzymes are mostly proteinaceous. Okay, but there are some enzymes which are also made of RNA, and such enzymes which are made of RNA, they are said to be ribosome. For example, 23S rRNA. 23S rRNA, this is an enzyme, and this is involved in the what? In the translation process. It is involved in the translation process, and this transfers the peptide chain from the P site of the ribosome to the A site. Okay, and this 23S rRNA is also called as peptidyl transferase. Why? Because it transfers the peptide chain. Okay, it transfers the peptide chain. So the example is what? 23S rRNA. Okay, and what do you call this as? We call this as peptidyl transferase. What does this do? It transfers the peptide chain from P side to the A side during translation. Okay, nucleic acid dihybrid cross, this is incorrect. Okay, so the answer we have already got. Th Morgan did not give the translation. Th Morgan gave what? Linkage concept. Okay, Mendel, he did not perform transformation. It was what? Griffith who performed transformation. Next, the final proof for DNA as genetic material came from the experiment. From the experiment of what? Hershey and Chase experiment. Okay, and they performed what? Transduction experiment. They used bacteria and they used bacteriophage for their experiment, and they found that okay, DNA is the genetic material without any doubt. So the answer is Hershey and Chase experiment. Next, the Taylor conducted experiments to prove semi-conservative mode of chromosome replication in. See, for the first time, the DNA replication is semi-conservative was proved by Michelson and Stoll, and they proved it in the E. coli. Then other scientists also proved this one, but in different organisms. So Taylor et al. He proved semi-conservative mode of DNA replication in which organism? Visia faba. This is a bean. This is a bean. Okay. Next, a molecule that can act as a genetic material must fulfill the traits given below, except it should be able to express itself in the form of Mendelian characters. Yes, it should be. Okay, but here the question asks about exception, so this is not the answer. It should be able to generate its replica. Yes, it is the criteria which should be fulfilled by the genetic material. It should be unstable. No, it should be stable. It should be stable. So the answer is C. It should provide scope for flow of changes that are required for evolution. Yes, this is also true statement, and this is incorrect statement. This is false statement. So the answer is C. Next, transformation was discovered by. Who discovered transformation? It was Griffith, who performed his experiment with the in mice, and he used the bacteria Streptococcus pneumoniae or Pneumococcus pneumoniae, and he performed transformation experiment. Okay, why? Because he found that the R form of bacteria got converted into the S form of bacteria. That is why he called that experiment as transformation experiment, because one form of bacteria gets converted into another form of bacteria. That is why transformation experiment. Next, the unequivocal proof of DNA as genetic material came from the studies on what? On virus. On the virus. And what virus? Bacteriophage. What is bacteriophage? Bacteriophage is that virus which uses bacteria as its host. So that is called as bacteriophage. Okay, next. Semi-conservative mode of DNA replication was first demonstrated in where? In which organism? E. coli. This is very simple question. Okay, transformation experiment was first performed in which organism? Transformation experiment was performed by what? Performed by Griffith. It was performed by Griffith in the year 1928. Okay, in which organism? Pneumococcus pneumoniae, which is also known as Streptococcus pneumoniae. Okay, Diplococcus pneumoniae. Okay, right. Next, and see, he used two varieties. What are those? Two strains. One was R2 strain and second was S3 strain. Okay, and this R2 strain is rough. Why it is rough? Because why it is smooth? See, rough it does not mean when you touch it you will feel rough. No, this is not the reason. You call a bacterial colony as rough if when you look at the bacterial colony, if they appear dull in color, then that is rough. If they appear bright, they are called as smooth. Okay, and what is the differences between R2 and S3? See, the R2, it does not have any capsule. So I will call this as non-capsulated. This is non-capsulated. And this S, it has capsule, so I will call this as capsulated. And this Pneumococcus pneumoniae or Diplococcus pneumoniae, it causes what? It causes pneumonia disease. Okay, and for causing the pneumonia disease, there must be some chemical present in the in this bacterium, right? So where do you find this chemical which is responsible for causing the pneumonia? It is the capsule, and because capsule is present in this S3, that is why the chemical is present for pneumonia, and that is why this will cause pneumonia. So I will call this as pathogenic. This is pathogenic, and these pathogenic forms are also, for pathogenic we also use another term, that is virulent. We use the term virulent. Whereas because the R2 form, it does not have any capsule, so this is non-pathogenic, and this non-pathogenic can also be called as what? Non-virulent. Non-virulent. Okay. Now next, pneumococcus experiment proves that bacteria do not reproduce sexually. RNA sometimes control the production of DNA and proteins. DNA is genetic material. Bacteria undergoes binary fission. See, pneumococcus pneumonia experiment. That means what? That means transformation experiment. Which experiment? Transformation experiment. Transformation experiment. This question is little bit incorrect. Why? Because this did not say that DNA is genetic material. Later on, with the help of this experiment, we concluded, okay, DNA is genetic material. Okay, who proved this? It was Avery, McLeod, and McCarthy. Okay, so but because no option is perfectly correct, so I will write this one as correct. Okay. Now next, DNA synthesis can be specifically measured by estimating incorporation of radiolabel. See, DNA has a special nucleotide or nitrogenous base, which is thymine. Okay, so so if I want to label the DNA, I will not use radiolabeled uracil. Why? Because uracil is never present in the DNA. I will not use adenine, radiolabeled adenine. Why? Because radiolabeled adenine is also present because adenine is present in both DNA and RNA. It is present in both DNA and RNA. Okay, uracil is present only in RNA. So because adenine is present in both DNA and RNA, so I will not use radiolabeled adenine to label the DNA molecule. Why? Which one we want to label? Only the DNA molecule. So I will not use adenine. Why? Because it is also present in RNA. So if I use radiolabeled RNA, then radiolabeled adenine. So what will happen? Both DNA and RNA, they will get radiolabeled. If I use radiolabeled uracil, only RNA will be radiolabeled. If I use deoxyribose, I can radiolabel the DNA. If I use thymidine, I can label the DNA molecule. So among these two, we can use both thymidine and deoxyribose sugar to label the DNA. So among these two, which one should I use? I should use thymidine. Why? Because the amount of thymidine is less and the amount of deoxyribose sugar is more. So if I want to label properly a DNA molecule, I should use the thymidine, because less amount of thymidine is sufficient to label the DNA molecule. So the answer is thymidine. Next, the transformation principle of pneumococcus as found by Avery, McLeod, and McCarthy was. Okay, so Avery, McLeod, and McCarthy, they performed an experiment to characterize the chemical nature of what? Transformation principle, and they found that the genetic material was DNA molecule. Just a minute. So how did they found the genetic material was DNA? So see, they took heat killed S3. Okay, so from heat killed S3 they isolated chemicals. So I will say chemicals isolated from heat killed S3. And they purified it. And after purification, they distributed them into three groups. What were those? One was DNA. So they purified DNA over here. Then they purified RNA and they purified protein. They also purified what? They also purified lipid. So we will not consider lipid over here. Okay. So the DNA was divided into two groups. In one case, only DNA was taken, and in another case, some amount of DNA was supplied with DNase. And what is DNase? DNase is the enzyme which digests the DNA molecule. And the RNA was divided into two groups. In one case, only RNA was taken. In another case, some amount of RNA was taken, and it was supplied with the RNase. Which will digest the RNA molecule. And here, the protein was divided into two groups. In one case only pure protein was taken, and in another case some amount of protein was taken along with the enzyme protease. Enzyme protease. Okay, now what did they do? They had taken culture medium. So they took culture medium. So this is one culture medium. So they had taken culture medium. They had taken culture medium. I need just 1 minute. Okay. Okay. Now, in this culture medium, they had taken only R form. They had taken only R form. They had taken R form of bacteria. R2 form of bacteria. Which form of bacteria? R2 form of bacteria. Which bacteria? Diplococcus pneumoniae. Diplococcus pneumoniae. Okay. Now, what did they do? They incorporated the DNA molecule into the culture medium, and they found that after some time, here the R form bacteria are present, and apart from R form of bacteria, S form of bacteria are also present. That means what happened? Some of the R form of bacteria got converted into S form. So I will say the transformation has taken place over here. Okay, in this case, what happened? In this case, only R form of bacteria were present. Only R form of bacteria was present. That means here what did not happen? The transformation did not happen. Here in this case, what happened? Here only R form of bacteria was present. Only the R form of bacteria was present. Here also only R form of bacteria was present. Here also only R form of bacteria was present, and here also only R form of bacteria were present. That means in all these cases, just a minute. In all these cases, what happened? You can see that in these cases there is no transformation. In this case, no transformation. Here also no transformation. Here also no transformation. Why? Because only R form is present. But in this case, some of the R forms got converted into S form. Why? Because you added DNA molecule. So you can see that because when we added DNA molecule, then some of the R form got converted into S form. That is why I will say DNA is responsible for changing R form into S form. That means that is responsible. That is the transformation principle, and the transformation principle proposed by Griffith was actually genetic material. So from this they concluded, okay, DNA is genetic material. In this case, what happened? They had supplied DNA molecule. But along with DNA, they had added DNA. So the DNA is broken down. That means in this the mixture which was added to this culture medium did not have any DNA molecule. Why? Because the DNA broken down the had broken down the DNA molecule. So that is why no transformation took place. Here RNA molecule was added. So RNA molecules was added. There was no transformation. When RNA plus RNase was added, that means when you add RNase plus RNA, that means RNA will not be there. That will be broken down. Here there was no transformation. Here also no transformation. Here also no transformation. So by comparing all these, they concluded, okay, DNA is the transformation principle, which was proposed by whom? Griffith. Next, who proved DNA is basic genetic material? Who proved DNA is basic genetic material? It was what? It was Hershey and Chase. Okay, Watson did not prove. He was associated with giving the double helical structure of DNA. Okay, Griffith, he did not say anything about DNA molecule. It was his failure. It was his drawback of his experiment. He could not provide the chemical nature of the transformation principle. So Griffith won't be the answer. Okay, Watson won't be the answer. Boveri and Sutton, they gave what? They gave chromosomal theory of inheritance. So this is not the answer. So the answer will be D. Next, E. coli fully labeled with N15 allowed to grow in N14 medium. 2 strands of DNA molecule of the first generation bacteria would have. See, suppose this is one DNA. And in this DNA, the nitrogen are N15. That means heavy nitrogen are present. Which nitrogen are present? Heavy nitrogen are present. And after replication, I will get DNA like this. Here, what did you supply? You supplied N14. You have supplied what? You have supplied N14. Okay, N14. So these are the two DNA molecules. These are the two daughter DNA molecules. Okay. So here it will be N15. Here it will be N15, and this will be N14, and this will be N14. Okay. So the density of this DNA will be more than the density of this DNA and density of this DNA. Okay. So the density will be different. The density will be different, but the sequence of this DNA will be exactly identical to the sequence of this DNA. Okay. So I will say that different density and do not resemble with parent DNA. No, they resemble with parent DNA. Why? Because this strand will resemble with this strand. Okay. And this strand will resemble with this strand. Okay. So here because it is written that do not resemble, so this is incorrect. Okay. Different density but resemble with parent DNA, this is correct. Same density, different density, same density is not correct, so the answer will be B. Next. Which one of the following is not applicable to RNA? See, not applicable. Heterocyclic nitrogenous bases are that? Yes, of course, because RNA has pyrimidine. RNA has pyrimidine, and which pyrimidines are there? Cytosine and uracil are present, and the pyrimidines are heterocyclic. They are heterocyclic. We have already discussed about this. Okay, so this is true, and we have to find out not true. Chargaff's rule, yes, this is not applicable, so the answer will be B. Complementary base pairing. Complementary base pairing, yes, it can take place in case of tRNA. When you draw the diagram of tRNA, so suppose this is tRNA. Suppose this is tRNA, this is 5' and this is 3'. These are the loops, okay, and these are the arms. So in these arms there will be hydrogen bonds. And these hydrogen bonds is due to complementary base pairing. So in RNA, complementary base pairing will be there. 5' phosphoryl and 3' hydroxyl group, yes, in this end phosphate will be there, in this end hydroxyl group will be there. So this is also true, so this is true, okay, and this is also true, so the answer is B. Okay, next. Similarity in DNA and RNA is that both are polymer of nucleotide, both have similar pyrimidine, both have similar sugar, both are genetic material. No, both are polymer of nucleotide, both have similar pyrimidine. No, this is incorrect. Why? Because in DNA, if you talk about the pyrimidine, then the pyrimidine in DNA are what? Cytosine and thymine, and pyrimidine in RNA are cytosine and uracil. So they are not similar, they are different. Both are similar sugar. No, if you talk about the sugar, sugar in DNA and sugar in RNA, they are different. Why? Because in DNA the sugar is deoxyribose. Right, and in RNA the sugar is ribose. So similar sugar, this is incorrect. Both are genetic material. No, in normal cases, when we talk about the organism, we talk about cellular organism. In cellular organism, the genetic material is DNA, not the RNA. Both are polymer of nucleotides. Yes, both of them are polymer of nucleotides. So I will say A. C. RNA can also be genetic material, but in RNA viruses and viroids. And whenever we talk about genetic material or think about organisms, we have to think with respect to cellular organisms. In all cellular organisms, the genetic material is always double-stranded DNA. Okay. Now, during DNA replication, Okazaki fragments are used to elongate. See, if I draw a diagram of replication fork, so suppose this is one replication fork. Suppose this is one replication fork, and suppose this end is 3' end and this is 5' end. This will be 3' end and this will be 5' end. Okay, now this is one Y-shaped structure. This will be called as what? Replication fork. And in this case, what happens? There is one primer. there is one primer, okay, and this primer is elongated to form one chain, and this strand is this strand is called as what? Leading daughter strand. This strand is called as leading daughter strand. This is called as leading template strand. And in this case, what will happen? A number of Okazaki fragments will be there. So suppose this is the primer, this is the primer. So I'm drawing only two Okazaki fragments over here, okay. And now let me draw the DNA molecules. So this is DNA molecule, this is DNA molecule, okay. So you can see that the synthesis of DNA molecule is away from the replication fork. The replication fork moves in this direction, and the Okazaki fragments are synthesized in this direction. So I will say the replication fork is, the Okazaki fragments are synthesized away from the replication fork. Where? And this strand is called as what? Okazaki fragments, and this entire complex, entire strands will be collectively called as lagging daughter strands. So lagging daughter strands are synthesized in the form of Okazaki fragments away from the replication fork. So I will say lagging strands away from replication fork. So the answer will be C. Next, method of DNA replication in which two strands separate and synthesize new strands, and that is called as semi-conservative. What do you call that? As semi-conservative, okay. So what is semi-conservative, conservative, and dispersive? Let me tell you. So if in semi-conservative, what happens? So suppose these are the two strands, they get separated, and after separation, what will happen? New strands will be synthesized. So this one will be used as a template. So using this one as template, new strands will be formed, and using this one as template, new strand will be formed, okay. So these are the two daughter strands, two daughter DNA molecules, okay. And this is called as what? Semi-conservative. Why do you call this as semi-conservative? Because in each daughter DNA molecule, this is one daughter DNA molecule, in each daughter DNA molecule, half of the DNA of the parent, if I take this DNA, this one strand of this DNA, that means I'm taking half of this DNA. So I will call half of this parent DNA is conserved over here. So I will call this as semi-conservative. So what is conservative? In case of conservative, what happens? So suppose this is the parent DNA. And after replication, what will happen? I will get two DNA molecules, and in one DNA molecule, the entire parent DNA will be present. That means the 100% of this DNA is present in the daughter DNA molecule. So it is conservative, and one DNA will be completely new. Okay. So this is called as conservative. What is dispersive? In case of dispersive, what happens? In case of dispersive, let me draw this one over here. So suppose this is parent DNA, this is parent DNA, okay. And now what will happen? I will have daughter DNAs. So after replication, I will have daughter DNA molecules. So this is newly synthesized part, this is newly synthesized, okay. Now so in this case, you can see each strand of the daughter DNA consists of both parent segment and newly synthesized segment. So this type of DNA replication is called as dispersive. Okay. Next, during DNA replication, the strands separate by. Which is responsible for separating these strands? It is the unwindage or helicase. Okay. How does this do? See, this unwindage has ATPase activity. So it will hydrolyze the ATP, and using the energy released from the ATP, it will break the hydrogen bonds present between the two strands. Okay. And once the hydrogen bonds are broken, the two strands will get separated. Okay. So the answer will be unwindage or helicase. And this helicase enzyme is also called as DNA B protein. This is also called as DNA B protein. And what does this enzyme has? It has ATPase activity. So it will use ATP to break the it uses ATP to break the hydrogen bond. So I will say it uses energy released, or it uses ATP to break hydrogen bonds between two strands of DNA. Two strands of DNA. Okay, next. Experimental material in the study of replication has been, see, the experimental material which is used to study the DNA replication, that means how the DNA replication takes place, what is the mechanism of DNA replication, it is the E. coli. It is the E. coli. DNA replication is conservative. No, it is semi-conservative. It is semi-discontinuous. So I will say semi-discontinuous and conservative. Why do I call this as semi-discontinuous and semi-conservative? Because when we form the daughter DNA molecules, in each daughter DNA molecule, one strand is parental, one strand is what? Newly synthesized. That is why I call this as semi-conservative. I can write over here why semi-conservative, because if this is parent DNA, and suppose these are daughter DNA molecules, okay, so these are daughter DNA molecules. So you can clearly see that in these are the daughter DNA molecules. This is daughter DNA, this is daughter DNA. In daughter DNA, one strand is newly synthesized, one is parental. One strand is parental, one is newly synthesized. That means this is semi-conservative. Why? Because in each daughter DNA molecule, half of this parent DNA is conserved. So this is semi-conservative. Why semi-discontinuous? Why DNA replication is semi-discontinuous? Because if you talk about the mode of DNA replication, so suppose this is one replication fork. Suppose this is one replication fork. So in this replication fork, so suppose this is 3' and this is 5' and this is 3' and this is 5' end. So in this case, what will happen? One, suppose this is primer. This is primer. Suppose these are the primers. These are the primers. Okay. And in this case, what will happen? This strand is synthesized continuously. This strand is synthesized like this. This strand is synthesized like this. This first strand is synthesized like this. So you can see one daughter strand is synthesized continuously. Another is synthesized discontinuously. That is why this is called as semi-discontinuous. Semi-discontinuous. Okay. Next. Name the enzyme that facilitates the opening of DNA helix during transcription. So during transcription, there is no helicase enzyme involved. During replication, the opening of the DNA helix is by the help of helicase enzyme. But during transcription, there is no helicase. So who is going to open the DNA molecule or the break the hydrogen bonds? It is the RNA polymerase itself. So the answer is RNA polymerase. The answer is RNA polymerase. Next, match the column. Okay, DNA polymerase first. We know all of us know very well that in prokaryote there is a single type of RNA polymerase which synthesizes all the three types of RNAs. So I will say in prokaryote one, that means one RNA polymerase, single type of RNA polymerase, it synthesizes three types of RNA, all the three types of RNA. That means RNA, mRNA, rRNA, tRNA, all are synthesized by the RNA polymerase. But in case of eukaryote, the RNA polymerase is of three types. Okay. So what are those RNA polymerase? So I have RNA polymerase first, RNA polymerase second, RNA polymerase third. Okay, and they will synthesize different type of RNA. So RNA polymerase 1, it synthesizes different types of ribosomal RNA, except rRNA, except 5S rRNA, RNA polymerase second, it synthesizes the hnRNA. hnRNA is also known as pre-mRNA. Okay, and RNA polymerase 3, it synthesizes tRNA. And 5S rRNA. 5S rRNA is also synthesized by what? The RNA polymerase 3. Okay, so the answer will be RNA polymerase first. Okay, so this is RNA. Okay, so I will write over here RNA polymerase first is what? rRNA. So one will be, this is 1, this is 2, this is 3. Okay, so 1 will be 2, 1 will be 2. Okay, so this is the answer. 2 RNA polymerase second is hnRNA. So this is 3, 2 is 3, 2 is 3, and 3 RNA polymerase 3 is tRNA. So this is the answer. This is the answer. Okay. Next. The sequence of coding strand is given. The sequence of coding strand is given, and it is asked that what will be the sequence of mRNA. Remember one thing, the sequence of mRNA and sequence of coding strand is similar. There is, except in DNA it will be thymine, in RNA it will be uracil. So if we are given the sequence of coding strand, we can find out the sequence of mRNA. Okay. So what will be the sequence? Here A, so here also it will be A. Here also A will be there. Here GG, here also GG will be there. Here T is present, here U will be present. Here AT, so here A, here U will be present. Here CA, and here T is present, so here it will be U. Here it will be U. So the answer will be A. What is the key to answer such questions? The key is this sequence, nucleotide sequence of coding strand and the nucleotide sequence of mRNA is similar, except in DNA it will be T, in RNA it will be U. Okay. Next. Next. Which of the following RNA should be most abundant in animal cells? See, in animal cell or eukaryotic cell, we have different types of RNA. Messenger RNA is there, ribosomal RNA is there, tRNA is there. Among these 3 RNA, which one is the most abundant one? rRNA. Ribosomal RNA is the most abundant. So the answer will be ribosomal RNA. Spliceosome do not found in. See, spliceosome is responsible for splicing. This is responsible for splicing. And what is splicing? Splicing is a process in which the introns are removed and the exons are joined in a sequence from in the hnRNA to produce the mature mRNA, and this process occurs only in eukaryote. Only in eukaryote. So I will say what is splicing? Splicing is removal of what? Introns. Introns are removed and joining of what? Exons in a sequence. In a sequence. In a sequence, and this occurs only in eukaryote. This occurs only in eukaryote, not prokaryote. And because bacteria is a prokaryote, so the answer will be C. Why? Because not found. Not found. Okay. So animal cell, it is eukaryotic in nature. Fungus, it is eukaryotic. Plant is also eukaryotic. So the answer will be C. Next, the equivalent concept, the equivalence of a structural gene is what? The equivalent of a structural gene is what? Let me tell you. These terms. Mutant. What is mutant? Mutant is the unit of mutation. That means this is a segment of DNA which undergoes mutation. That is known as mutant. What is operon? Operon is a segment of DNA which undergoes independent expression and its regulation. That is known as operon. What is racon? Racon is a segment of DNA which undergoes what? Recombination. That is known as racon. So what is cistron? Let me tell you. There is a term. So what is the similarity between gene and cistron? Let me tell you one by one. So suppose this is a DNA molecule. Suppose this is a DNA molecule. Okay, and I am taking some segments over here. So suppose this is one segment, this is another segment, and this is another segment. So I have taken 3 different segments. Okay, so this segment, suppose this is segment 1. Okay, and this segment underwent transcription to produce the rRNA. This underwent transcription to produce the mRNA, and this segment underwent transcription to produce the tRNA. So these are the segments of DNA, and they have functions. What is their function? To produce different RNAs. So they are said to be genes. So suppose this is gene 1. Suppose this is gene 1, this is gene 2, and this is gene 3. So what is gene? Gene is basically functional segment of genetic material or DNA. Okay, so this segment has a function to produce the rRNA. This segment has a function to produce the mRNA, and this segment has the function to produce the tRNA. Okay, so most of the students, they think that gene is basically the segment of DNA which produce protein only. No, it can also produce the rRNA, it can also produce the tRNA, and if it produces mRNA, ultimately it will undergo translation to produce the protein. It will undergo translation to produce the protein. So what is cistron? In this case, this segment, and this segment, all these segments are genes. So I will say this segment A, this segment B, and this segment C, all are genes. But if you talk about cistron, then only this segment B is cistron. This is not a cistron. This is not a cistron. So I will say only this one is cistron. So what is cistron? Cistron is the segment of DNA which produces protein. That means which produces mRNA. But what is gene? Gene is a segment of DNA which can produce either rRNA or mRNA or tRNA. That means I can say from this, I can say that all cistrons are always genes, but all genes are not proteins. So I will say all cistrons are genes, but all genes are not cistrons. And cistrons are of two types, monocistronic and polycistronic. Okay, so monocistronic condition is found in eukaryotic cell and polycistronic condition is found in prokaryotic cell. So hope you have understood some of the concepts. Okay, so we will and I hope you have enjoyed this class. So take care and study well. Thank you.